Thank you very much for the introduction, Philip. Yeah, as he said, my name is Markus Orsi. I studied pharmaceutical sciences at ETH Zurich. And in last September, I started my PhD in cheminformatics at the University of Bern in the Raymond Group. And today, I'm actually going to talk about how we use machine learning in the discovery of membrane-disruptive non-hemolytic anticancer peptides. I would like to start by telling you why we are actually interested in this class of therapeutics. So for those of you who don't know, membrane-disruptive peptides are often very short peptide sequences, around 5 to 25 residues, and they often take on this alpha helical amphiphilic structure, which enables them to insert themselves into biological membranes, such as bacterial ones, or cancer cells ones, like in our case. And the nice thing about this mechanism, due to this mechanism, basically these peptides kill the cell that they can insert inside the membrane, and they kill the cell in the process. Because of this, the cells, both bacterial and cancer, very often do not manage to develop drug resistance, which is quite nice, considering that this is a growing problem. Also, because of their peptidic nature, they often display low immunogenicity, good biocompatibility, low metabolic toxicity, since especially when we are using only natural amino acids, this is not a problem generally. And they're also very easy to synthesize. The only major issue of membrane-disruptive peptides is that they often lack selectivity for our target membranes here. So they very often attack also healthy human cells, most specifically red blood cells, which is why these therapeutics are very often displaying hemolytic effects. So they kill red blood cells, which is the reason why when we design such sequences, we're not only interested in designing active sequences, but also non-toxic ones. So I briefly show you the workflow, and then I will go more into detail. So we start with the peptide design, and here we generate a virtual peptide library, after which we then use an activity classifier to filter out inactive sequences, or sequences that are predicted to be inactive. We use additional selection filters and then synthesize and test them in the lab, actually. And then finally, we also do some post-hoc analysis just to gain further insight into our experimental results. Just at this point, I would like to thank Dr. Lice Capecchi, who's the one that worked on all of the machine learning models that I'm showing today, and Elena Zakharova, who has synthesized and tested all of the compounds that I'm showing you. So starting with the peptide design, here we use a recurrent neural network that is composed of 2 layers of GRU cells, and it's actually trained on 9,548 linear sequences from the database of antibacterial activity and structure of peptides. This step was mainly aimed to make the generative model actually generate sequences that are not random, but that at least resemble what humans have made already. Because as also for those that attended previous talks, the peptide space is just huge. So the combinatorial space is just enormous. So this is why we actually try to focus on sequences that resemble these ones. And in a second step, then we actually do the transfer learning with 53 peptides active against HeLa cells. These are cancer cells, human cancer cells. And this should guide the generation of our sequences towards sequences active against cancer. From this exercise, we sampled a total of 50,000 sequences. And we applied then our activity classifier on all of the 50,000 sequences. So you can see the architecture is quite similar, but the output is either a sequence is predicted to be active or inactive. Here we had trained it, again, always the same database, but this time only on activity data of a bit less sequences. So here sequences were either labeled to be active or inactive. We actually borrowed this model from a previous publication where we aimed at making antibacterial peptides. And here you see that in this publication we also compared our RNN with other classifiers. So we actually just took the best one that we had, which was the RNN here. And so after filtering out all the sequences predicted to be inactive, we moved on and we applied additional selection filters. So the first one I would like to talk is the Levenshtein distance from the training set and the test set. For those that don't know, the Levenshtein distance is basically when you compare two sequences, the amount of insertions, deletions, and mutations that you have to do on one of the sequences so that they are the same. And we chose this filter just because we did not want to get sequences that were too similar to what we already showed to our model. And we also discarded sequences with a Levenshtein distance higher than 8, just to remove, so because this was just outside of the applicability domain, so the results, so our models would, the results of the models would not be in applicability domain, yeah. So an additional filter that we used was the predicted helicity and hydrophobic moment. These are just physical properties of these sequences. I showed you they're often alpha helical, and they also need a certain amount of hydrophobicity to be able to insert into membranes. So we just removed those sequences that did not display such properties. Just for the ease of synthesis as well, we kept sequences with a maximum length of 15 amino acids and also only L amino acids. This was purely just for synthesis reason, just to make it a bit easier. After all these filters, we end up with 202 sequences on which we clustered using the Butina algorithm. And we obtained 14 clusters, and we synthesized then, or we selected then for synthesis the 14 centroids of the clusters, but the synthesis of one didn't work. But here you actually see the experimental data of our synthesized sequences. So these are not actually the predictions, this is the actual experimental data, and you can see that for cancer cells, we already have 7 out of 13 sequences that are active against these HeLa cells. But since also all of the machine learning models that we used were borrowed from this previous antibacterial project, we also tested our sequences on bacterial cells, and here also we see that most of them are active even against Acinetobacter baumannii. It's not really surprising, these sequences very often share activity among different types of cells, which is also the reason why most of the time they're toxic, because they just kill everything. What we were also quite surprised or happy to see is that most of these sequences are actually helical, so this kind of corresponds really to what we are expecting that they would, or how they would behave. But I told you in the beginning that we're not really interested, or we are obviously mostly interested in having active sequences, but we're not exclusively interested in activity. We also want to have non-toxic peptides. And for this reason, actually you also tested hemolysis, of course, but we also wanted to include a hemolysis classifier into our workflow. So basically, what we did was very similar to the activity classifier. We trained a second model with a very similar architecture, but just one layer of GRU cells, and this time on hemolysis data, always from the same database, to kind of predict if a certain sequence is possibly hemolytic or not. Again, we compared this with other classifiers, and we found that the RNN was actually the best one. So we applied this classifier on our 13 synthesized sequences. You see the prediction here and the experimental value here. So actually we see that 7 out of our sequences are non-hemolytic, but that, let's say, the prediction was correct in 8 out of 13 cases, which if we ask someone from machine learning or data science might not be very convincing, but since these are actual experimental data and this is a really applied project, we are quite happy to see that most of the tiny predictions are actually correct. Since we, in the future, would like also to use this classifier before actually selecting and synthesizing our compounds to already filter out hemolytic sequences. But we were also curious just to see how the classifier behaves on a bigger set. So we applied it actually on the entire DBASP, so the database that I've been talking about all the time. We encoded all of the chemical structures of these peptides with the MAP4 fingerprint, which is quite similar, let's say, to ECFP ones, but it's just better suited also for displaying bigger sequences, bigger structures in general. You can see in the UMAP space here that there are a few clusters, but there is like no clear clusters. So this is actually quite a difficult task to carry out. This indicated us that it's not very trivial to define subspaces in the peptide space where all of the sequences are actually non-hemolytic. We also wanted to kind of visualize this with TMAP. This is a visualization algorithm that we use or that we have developed in our group and that we kind of use pretty often, especially to visualize large data sets like this one, because as you can see, it fills out the space a bit nicer. And you can actually see that here we have sort of quite a few clusters, and we think that this is actually what the classifier is actually getting out of what it's actually understanding about the database. So, but overall, we can see that this still remains a quite hard task. So we're really hoping that in the future, we can use a kind of improved version also of this classifier to also drive our selection for sequences and to not only have then active sequences, but also non-hemolytic sequences. And with that, I'm already finished. I speak very quickly and I don't like to share too many details. So I would just like to thank you for your attention, of course, and special thanks go to Prof. Jean-Louis Raymond, who kind of gave me the supervision, Dr. Alicia Capecchi and Elena Zakharova, who have made most of the work that I presented today, actually. The entire Raymond group and also the organizers of this track. And thank you for having me.
Thank you very much for a nice talk. Are there any questions?
So thank you for the talk. I just have a question. Why do you remove a GRU layer in the second model? Because you had fewer data points, right?
Yes. So honestly, since I haven't worked actually on this, I don't have an answer for it. I'm sorry.
Okay. Thanks.
Other questions? So I have a question about the T-maps slash U-maps you show.
T-maps, U-maps, yeah.
What descriptors are you used there to?
Exactly. So the descriptor is MAP4 actually. It's quite similar in how it will like to ECFP fingerprints, but it also includes measurement of how distant certain atom pair. So it's kind of a mixture between ECFP and atom pair fingerprints, which is why these kind of exact as the ECFP would be, but also let's say suited for bigger structures, like in this case.
Have you tried something closer to what your GRU model sees? So, for example, I don't know, n-grams on the amino acid sequences, or something similar, just to see if it's.
Yes, honestly, no, or at least we tried, but it didn't look. We could not kind of determine any meaningful display, basically.
So it's really challenging to separate.
It is. Okay. It is quite challenging. Yes.
Okay. Thanks. Other questions?
Thank you. Very interesting talk. So I also have a question about this representation. As I see, you showed here that basically this homolytic or non-homolytic property is spread in the space. But I wonder if you thought about other properties that could be maybe that could form some groups in this space.
Yes. So actually, when you color code by length, you see that this is kind of the main, let's say, the property which gives the main variance. So really, you in the UMAP space, you would see that all the short sequences are here and all the long sequences are up here, and the same in TMAP as well. Of course, we can do this also with activity. So I actually did. I actually did it also with the activity and also the activity and non-hemolytic. Quite often, the reason why we also like to use TMAP is that it works very well with another package that we have in-house, which is Faroon, where we kind of display all of these maps, even the UMAP, in a very interactive manner. So you I cannot show it on a presentation, unfortunately, but you can kind of add lots of categories to which you color, and then you can really find out which, let's say, properties give you a nice clustering in space or separate very well on this space. So yeah, there are others, I guess, but here in this specific project, I don't think I saw anything that was clustering very well, actually.